My name is Tom Thompson. I'm a professor at the University of Cincinnati in the Department of Molecular Genetics, Biochemistry, and Microbiology. The types of students that we get are very interested in understanding how biology works. And then they come across our lab and then they see how interesting structural biology is. We're a structural lab, and what we try to do is understand molecules, how they signal and how they function in the transforming growth factor beta family. These are a group of signaling molecules that number about 33, and they interact with cellular receptors to do a whole host of cellular responses and are involved in development to the adult. They're involved in neurogenesis, they're involved in immune surveillance, a lot of regeneration processes, wound healing, play a major role in a number of pathologies. For instance, they're misregulated in a number of cancers, a number of other diseases. One of our main techniques is called XRay Crystallography, where we express to high levels a particular protein of interest, and then we form crystals of that purified protein. We've solved many different structures in the TGF-beta family. We work on how these molecules interact with their cellular receptors. We also work on how these molecules interact with their protein antagonist. Another technique besides X-ray crystallography that we're really interested in doing is called CryoEM. This has really made some major breakthroughs in the last 4 to 5 years with resolution. We're very fortunate at the University of Cincinnati to have a facility that just provides our students with the ability to become trained in cryo-EM, gives them an opportunity to be sort of more diversified and increase their toolbox for structural biology. We also, in the lab, like to couple our structural data with other types of data. And we do a lot of protein engineering, a lot of modifications of these proteins to try and understand what pieces of the protein are important for their function or their interactions. And so we dissect a lot of the proteins that way. And then we use a lot of cellular assays to try and figure out what components of those proteins are important for their signaling, what makes them unique, and then how do they interact with other proteins. The types of students that really join the lab are ones that are pretty self-motivated. They really want something out of their career. So it is a lab where you're not micromanaged, where you have an independent project. And I work with you to sort of hone in on kind of what the goals are for that project. And at the outcome, the goal is to get a number of publications, to solve structures, to figure out how things are working. And really, we have a lot of opportunities, a lot of different projects in the lab that allow for some really cool areas of biology.